Welcome, everyone. Welcome to the IU Indianapolis Center for Translating Research into Practice monthly conversation series with our Scholar of the Month. And this month, we're very pleased to have with us Bill Sullivan. My name is Steve Vieweg. I'm the director of the center. It's a pleasure to welcome you here. I have some introductory things I'd like to share with you, so let me pull up my slides to make sure that we are able to see the things that I want to share with you. So, here we are on October 25th. Great time in Indianapolis, lots of things going on. We're glad you took out a moment of your day to be here with us. And if you know folks who couldn't be with us, we are recording this. It'll be available later. So we want to give a shout out to the founders of the Center for Translating Research into Practice, Charles Bantz, who's Emeritus Chancellor of the campus, and in particular, Professor Emeritus Sandra Petronio, who sadly passed away about 6 months ago. But Sandra was the brainchild behind the whole idea of translational research as something we should pay attention to here on our campus. And one of her ideas was that we would have these conversation spaces where we could take time out of our busy schedules and just chat with one of our translational scholars and dig deeper into their work and try to understand it better. So we're so glad we have these opportunities today. So a couple reminders. We're all familiar with Zoom, but while we're getting started, as many of you are, go ahead and be muted and have your cameras off, but there will be time for conversation, and we hope that you'll turn on your cameras and your microphones, and we'll engage in some conversation together. Of course, you're welcome to use the chat, and since we are recording, if you don't want to be seen, you don't have to turn on your camera, but we hope that you'll enjoy that. I already mentioned we'll be recording this for viewing later, and you will receive one of those pesky evaluations afterwards. Please take a moment to fill it out. Give us your feedback about what you thought and give us some ideas about the future planning for our events. If you're interested, you can get continuing educations for units for this event. You can just reach out to us and we'll let you know how to sign up for that. But for those that need continuing education, this is a great way to do it while you're learning more about translational research here on campus. You can follow us in various social media ways through Facebook, X, Instagram, and we also have a YouTube channel, which is where you'll find the recording from today's session. We want to throw a shout out. It's always time to encourage people to learn more about the Bantz Community Research Fund, and this is how we are able to support annually somebody on campus as our Bantz Community Fellow who is advancing their research to the next level through an intense year. And there's some funding that goes along with that, but it takes dollars and it takes other support. So if you know anybody interested in that, or maybe you have some opportunity to support that, just go ahead and scan this now or call us and we'll tell you more about it. But check out our website where you can learn more about the Bantz Community Fellows and Scholars and the great work that they're doing. ScholarWorks is an important feature of our work here on campus, and it's a partnership with the library. And Bill Sullivan is a prolific person on ScholarWorks. So you can see that if you go to the ScholarWorks page at the library, there's all kinds of articles access, free, easy access to his articles in various journals. But you can see he's got like 13 pages worth of articles on ScholarWorks. But this is an easy place for you to go find out more about his work, but also all of the scholars here on campus that participate in this program. So where'd my mouse go? Hello, mouse, come back. Here we go. So next, though, upcoming events just next week on Wednesday. If you've got time, it's not too late to sign up to come join us for our Fall Awards and Showcase. It'll be in the Campus Center on Wednesday, October 30th, from 3 to 5 in room 409 in the Campus Center. And it's where we will honor this year's Bantz Petronio Trip awardee and the Bantz Community Fellow. But we'll also meet several other of our campus translational scholars and learn more about them. It's always a great time, and you get to just meet people and have a few snacks. And then in November, we're scheduled to have for the Scholar of the Month, Elizabeth Nelson, who will share some of her work. So sign up for that. It's early, it's November 15th because obviously we have Thanksgiving that month. So join us, learn more about this at the website. But today you've come to converse with Bill Sullivan. We're so glad that he's here. He's from the School of Medicine and he's the Showalter Professor of Pharmacology and Immunology and Professor of Microbiology and Immunology. But he's also the author of Pleased to Meet Me, Genes, Germs, and the Curious Forces That Make Us Who We Are. And so it's a pleasure to invite him here today to converse with us and to lead us through some great thinking and opportunities. So welcome, Bill.
Thanks very much, Steve. It's a real honor to be here and to participate in this Trip Scholar series. Um, Knowing that I was presenting in late October, so close to Halloween, I could not resist going with the zombie theme, which is going to be the central feature of the presentation I give today. I'll go ahead and share my slides if you're done with all the introductory material.
Yes, it's your time. Let's get conversive.
Okay. All right, if there is a problem, I assume you'll tell me.
We're good.
I assume it looks good. All right. So I've been at IU School of Medicine now for 24 years, and I've been working on this parasite I'll tell you about today called Toxoplasma gondii. And it relates to the zombie apocalypse in ways that I will reveal very shortly. Um, but in addition to my time at IU, I've been working on this parasite ever since I was a graduate student, which takes us way back to 1994. So I'm quite familiar with this parasite by now, or at least I hope so. And we've opened up our laboratory with the slogan that this is where parasites come to die, because at the end of the day, we are hoping to identify new drug targets in this phylum of parasites so that we can better treat toxoplasma and a lot of its cousins that cause a great deal of morbidity and mortality in human populations as well as animal populations. So let me advance here. Okay, so there is no shortage of examples of zombies in popular culture. They have infiltrated, or maybe I should say they've invaded video games, books, comics, television shows, movie theaters, and almost everything you can think of. Plenty of games that you can play on your phone. It's safe to say that we humans are kind of obsessed with zombies. So to get on the same page, what do we mean more or less when we say zombie? Well, that's an undead creature, something that is technically dead but still walking around for some reason. And in some cases, doing some very violent and unusual things like cannibalism to living people. So zombies can be created a number of different ways based on what you see in Hollywood. Everything from like radiation leaks to pathogens and all sorts of other things. And of course, you can become a zombie if you are bitten by a zombie. So all sorts of examples in pop culture, but at the end of the day, what all this amounts to is that when someone becomes a zombie, they cease to become a free agent. They cease to become who they were, and they are basically doing the bidding of whatever force caused them to become a zombie. And this concept has been extrapolated from the ancient religion in Haiti, the voodoo religion, and has just gone off in many multiple different directions in literature and film. So we were going to run some polls here, but we don't have the apparatus to do that. So I just want you to think for a moment. Do you believe zombies are real? Are there examples of those either in the human population or in the animal kingdom? And the answer to that poll question is yes. And I'm going to focus on pathogens that can create zombies as a lead-in to tell you about some of the work we do in our laboratory. So the classic example of how pathogens can create zombies would be rabies. The rabies is a virus that is transmitted through saliva via a bite. So one animal can transfer it to another through biting. But how does that virus get its host to become aggressive and bite? So that's where the zombie part comes in. When an animal is infected with rabies, it ceases to become itself. It doesn't behave like it normally did when it was uninfected and becomes much more aggressive and exhibits a much greater capacity for biting. And this is all to do the virus's bidding. This is how the virus gets around from one animal to the next. It rewires the brain, if you will, or changes its chemistry in a way to make that animal much more aggressive and then conducive to viral transmission. So some other examples that you may be less familiar with is this horsehair worm. And what you're looking at in this petri dish is a cricket. So this horsehair worm is emerging from the body of this poor cricket. And it's hard to believe, but this entire worm used to be packed into the body of this poor cricket. So why do I call this a zombie? Well, crickets normally avoid water. They don't jump into rivers or ponds or anything like that because they can't swim. It's basically a suicidal move on the part of the cricket. But when they're infected with this horsehair worm, they become suicidal, meaning they will seek out water and jump into it and basically drown themselves. Now, why would the worm want to drive the cricket to exhibit that sort of suicidal behavior? The water is where the worm needs to go in order to spawn. So it basically it takes advantage of this host organism, the cricket, to get it to where it needs to go in order to reproduce. Here's another example of a roundworm parasite that infects ants. On the top, you see an uninfected ant, and on the bottom, you see one that's been infected with this roundworm parasite. So the first thing you clearly see when the ant becomes infected, its abdomen turns bright red. But there's a behavioral change that takes place too. This round worm somehow alters the ant's brain so that it leaves the safety of the forest floor and climbs up a bush or small tree and attaches itself to the top of a leaf. And then it hoists its bright red abdomen up into the air. Why would the ant do this? Well, the ant is no longer in control of its behavior. This is the bidding of the round worm, and it manipulates the ant in this fashion so that it looks like a berry. So birds which never touch uninfected ants will happily swoop down and inadvertently eat this infected ant because it is mistaken for food. It's mistaken for being a berry. The worm wants to get into the bird because the bird has a tremendously greater capacity to spread the parasite far and wide through bird droppings around a wide radius. So a very clever move on the part of this round worm to spread itself far and wide. Now, one of the pathogens that creates zombies is all the rage these days because of a popular video game and television series called The Last of Us. And there is a kernel of truth that is the premise for this show, which is basically there's a fungal pathogen that can turn humans into zombies, as you see illustrated here. And it ultimately takes over the human body and emits spores and can spread around. This is based on a true living fungus that, again, infects poor ants. So the way this cycle works is that ants on the forest floor pick up these fungal spores, and once they become infected, their behavior changes. They leave the safety of the colony and they climb up a small bush or tree and attach themselves to the underside of a leaf. And they just clamp their mandibles down into the flesh of the leaf and they ultimately die. The fungus will overtake their body. This is a real picture of what this fungus looks like after it is infecting an ant. It sprouts these stalks, which release fungal spores down onto the forest floor to infect additional ants, and the cycle repeats. So I want to reassure you very quickly that this could never happen in humans. There's no way this zombie ant fungus is going to transmit to humans, but The Last of Us is based on this cordyceps fungus, which is real. So what about you? Is there a pathogen that turns people into zombies? I'm here to tell you today that it's quite possible. There is a parasite that is transmitted by cats, all feline species, actually, not just Garfield and the average household cat, but panthers and lions and ocelots and all felines can transmit this single-celled parasite called Toxoplasma gondii. And I'm going to tell you about that more, but first I wanted you to think about this question. You know, most of you out there, I actually can't see you at the moment, but I imagine some people out there have maybe never heard of Toxoplasma parasites and the fact that they infect humans. So how many people would you guess are infected with this parasite? And the answer turns out to be more than 1 billion, which is a staggering number of people. So current estimates say that about 3 billion people on the planet carry this parasite in their brain or other muscle tissues, and they're probably not even aware of it. I want to go through the life cycle real quick to give you an idea how toxoplasma has spread to so many people across the planet. And I should say right at the outset that toxoplasma, in addition to infecting humans, can infect any warm-blooded vertebrate, as you see in this life cycle cartoon here. So we'll start with the cat because that's what we call the definitive host. What we mean by that is that the cat is the only organism where the parasite undergoes the sexual stage of its life cycle. So it's excreted by cats in their feces in the form of oocysts. And these are very sturdy containers, microscopic containers in the feces that are stable in the environment for at least a year, maybe longer. So they thoroughly have contaminated our land environment as well as water. They're resistant to salt water and they can obviously contaminate fresh water as well. So these oocysts are everywhere out in the environment, depending on the cat populations in the neighborhood. So when other animals pick up these oocysts, they accidentally ingest these things. The parasite infects all of those creatures. Again, almost any warm-blooded vertebrate, mice, birds, and animals that we sometimes see on a dinner plate. So another way we can catch the parasite is by consuming these animals. If we eat raw or undercooked meat, there's a form of the parasite called a tissue cyst, which houses these bradyzoites, okay? They can infect humans when they consume raw or undercooked meat. There is another tragic way that the parasite can transmit from one person to the other, and that's from mother to child. So if a pregnant woman becomes infected for the first time during her pregnancy, the parasite can cross the placental barrier and infect the fetus. This can cause abortion, stillbirth, or devastating birth defects. And I think I have a slide that illustrates that momentarily. So many different multiple routes of transmission. And that is why Toxoplasma is so successful in spreading itself around the world. It's also a stealthy infection. If you have Toxoplasma in your brain, odds are you did not become very sick when you first acquired the parasite. Cats also do not exhibit tremendously, powerful symptoms that would indicate that they're sick with Toxoplasma. The infection is lifelong because of this bradyzoite encysted form. Once it encysts in the brain or the skeletal muscle or the heart, the parasite is just kind of stuck there, presumably for the rest of the host's life. So what I'm going to show you now is what we look at when we view Toxoplasma in the microscope. We grow them. We grow this parasite on a monolayer of human fibroblast cells. And this kind of mimics what would happen in the body during an active infection with toxoplasmosis. So you're seeing basically a lawn of human fibroblast cells, and all these little football-shaped organisms are the individual toxoplasma parasites that are happily growing inside of these fibroblast cells. The parasite doesn't replicate outside of a host cell. So if you come back the next day, you'll see host cells that are just packed with maybe thousands of these tachyzoid parasites. Okay, they're just literally packed. And you can imagine the next thing that's going to happen is a cell like this is just going to burst open, and all these little parasites inside are then going to invade neighboring cells. and start growing new vacuoles all over again. So if you come back another day, you will see that this very beautiful human host cell monolayer has been utterly destroyed by the parasites. You see some debris, but all these little banana-shaped organisms are the Toxoplasma parasites now able to infect new host cells. And like I said, you got to come into the lab and pass these pretty soon because they will start dying outside of the confines of a host cell. So when they lyse out like this, we got to pass them into fresh cells pretty quickly. And this usually happens, this lysis event usually happens, according to the folks in my lab, always on weekends and holidays, forcing them to come in and take care of these little parasites. But this is what I saw back in 1994 as I was being introduced into a parasitology lab. I had never heard of Toxoplasma before, but once I saw these pictures and how quickly it was ripping through cells, I was just utterly fascinated and had to learn more about this organism, and I've been studying it ever since. And you can imagine the devastating toll this can take on the body during an acute infection. If this were your brain or heart tissue, it can be destroyed very quickly. This is a new image that was generated by a talented postdoc in my lab, Vanessa Severo, and she's been developing a neuronal system so we can grow toxoplasma in the laboratory in neurons, the most physiologically relevant cell type where we want to study toxoplasma. So these are human neurons that are grown into a sphere, almost like an organoid. And all of these green dots that you see are toxoplasma cysts. These are the cysts that are located in the brain of infected people. And now that we have a culture system that Vanessa has developed, we hope that we can interrogate a little more precisely what the interactions between parasite and neurons are and throw drugs at this system so we can identify newer compounds that might be able to eradicate these cysts from the brain and body, which we currently cannot do. So toxoplasmosis as a clinical disease, it's usually seen as an opportunistic infection. Like I said, about 3 billion people are infected with this organism. They carry it, but they don't exhibit any symptoms of disease. And that's because the parasite is in its latent form. Now, it can reactivate, and this is what happens when the immune system becomes severely depressed, as you can see in AIDS or organ transplantation, sometimes in cancer chemotherapy. This is a brain scan of a patient with advanced HIV, and you can see that toxoplasma has literally eaten a hole in the brain next to the ventricle here, and this causes very devastating neurological effects and ultimately death if it can't be driven into remission by the current drugs we have. As I said before, there's congenital toxoplasmosis as well. The parasite can cross the placenta. It can cause hydrocephalus, encephalitis, which can lead to seizures in newborns. Many congenitally infected infants suffer from vision impairment like blindness and have intellectual disabilities as well. And then a newer front of research that emerged over the past 20, 25 years are reports of neurological problems. So even if people or other animals aren't exhibiting symptoms of toxoplasmosis, this latent form that insists in the brain, it looks like this, may be causing neuropsychoses or behavioral changes. And this circles me back to the zombie aspect of the talk. So there are studies that show that in humans who are infected with toxoplasma, they are twice as likely to develop schizophrenia. There are also well-controlled studies in rodents. Rodents who are chronically infected with toxoplasma exhibit behavioral modifications, and I'll explain some of that in a moment. So like I said before, I'm not making this up. You can go check out the articles if you like. People infected with toxoplasma have almost 2 times a greater risk of developing schizophrenia. And even people who own cats have 2 times greater risk of schizophrenia. So 2 different ways of looking at the same question, and they pretty much get the same result. So in addition to serious diseases like schizophrenia, there are some bizarre behavioral changes that take place. Like I alluded to in the last slide, rodents exhibit hypermotility. They tend to take more risks, including being unafraid of cats. which when you think about it makes evolutionary sense. As a rodent, they're a major prey of feline species. So if Toxoplasma finds itself stuck in a rat brain, the best thing it can do is change that rat's behavior to be unafraid of cats. That way, the rat is more readily consumed by the definitive host, and Toxoplasma gets into the gut of a cat where it really wants to go to complete the sexual stage of its life cycle. So this was met many years ago with a lot of skepticism, but has been reproduced in many independent labs across the world and appears to be a very real effect. It's even true in other species. So chimpanzees, for example, a non-human primate, they also they lose their aversion to leopard urine. Uninfected chimpanzees do not like the smell of a predator's urine, a leopard's urine. But if they're infected with Toxoplasma, they seem to be okay with it. Hyenas approach lions more closely. Wolves are more likely to be the leader of the pack, which echoes this risk-taking behavior that's seen in infected rodents. So what about humans? Well, a whole bunch of correlative studies have been done showing that risk-taking also tracks with people who are infected with Toxoplasma. They're also more prone to rage disorder. They're more likely to be involved in a car accident. And they're also more likely, interestingly enough, to be entrepreneurs, which again might harp back to the risk-taking tendency. So with a postdoc, I designed this shirt a few years ago that says, "Don't look at me, it was my Toxoplasma talking," as just kind of a tongue-in-cheek reference to the fact that if Toxoplasma does get into our brain, and even though it doesn't seem to be making us sick, it could still be influencing our personality and behavior. And I just find that absolutely fascinating and somewhat terrifying. So my lab is trying to get this parasite out of our heads because there's no cure at the present moment. There are antifolate drugs that can drive acute infection into remission, and those can be life-saving, but it doesn't eradicate the parasite from the body. So my lab is trying to identify new drugs, new drug targets that can eliminate these brain cysts. And we think we hit on one several years ago. It's an old blood pressure medication, believe it or not, called guanabens. And what you're looking at in this piece of data, and I'm not going to show you a lot of data, so don't get upset or tune out. But this is what a healthy cyst in vitro in a culture system looks like. Here we have our fibroblast cells again, and a nice healthy cyst growing here, just packed with parasites. If we treat that cyst with the guanabens, the parasites inside these cysts start to shrivel up and die, which was really cool for us to see because there's not a lot of drugs out there that have been documented to have this sort of activity. We put it into chronically infected mice and we saw a similar effect. We saw about a 70 to 80% reduction in the brain cysts that were treated with guanabens. Now, guanabens is particularly exciting because, as I said, it's already FDA approved for other medical conditions. So it could be rapidly repurposed as a drug that could reduce toxoplasma cysts in the brain. So with that, I just want you to think about your concept of free will. Now that I told you that there are pathogens out there that can modify mammalian behavior. So it might get you to evaluate that. And this is a hotly contested issue in neuroscience today, but the answer is no. And should we cure latent toxoplasmosis if it might alter personality? And I don't have an answer for this, but it does raise an interesting question that if toxoplasma can fundamentally change someone's personality, maybe it makes them an entrepreneur, for example. If we treat that person and get rid of the parasite, would it make them a little more cowardly? I don't know. It's an interesting question to contemplate. So the last two slides, which I understand will be distributed to everybody, just contain my contact information, ways you can continue reading anything that I write. You can follow me through these sites or social media. And there's a couple articles that I've written recently that I want to highlight, one of which is reversing the behavioral changes that Toxoplasma can create and Toxoplasma prenatal testing. So you might be a little surprised that given the high seroprevalence of Toxoplasma, and the fact that there is a test available for it. The US is kind of unique in among some countries in the world in that we do not test pregnant people for the presence of this parasite, which, like I said before, can cause stillbirth or congenital disease. So I wrote an article addressing that. And as Steve alluded to earlier, rooted in all of this work is the concept that we may be doing things that are beyond our control and may be under the influence of things that are under the hood that we don't appreciate. So that's what my book is all about. Pleased to meet me. It's about genes and germs like Toxoplasma and our microbiome and other curious forces that make us who we are. So if you like this subject, I think you'll really enjoy the book. So with that, I'm going to stop the presentation part so we can get on with the conversation. And again, I just want to thank everybody for taking time out of their day and tuning in. I really appreciate that.
Thank you for taking the time to share this information. Wow, it's a lot of things to think about. But I personally appreciate the way you were able to share it for somebody who is a not science kind of guy. But it raises a lot of questions. So this is the time to turn on your cameras and your microphones. And if you have a question or want to have some discussion, we'll open up the floor to ask and to talk about this. And we can also go back to some of the questions you posed if that's what we need to do. But well, let's open the doors and see if there's anybody here that has a question.
Bill, this is Gerardo. Quick question. First and foremost, your presentation is just fascinating. It's
Thank you.
really, really very nice, and you have made a very clear presentation of what the situation is. I am not a friend of cats. I don't like cats. I'm a dog person. But still, the issue here is there any consideration of reclassifying cats in a different category as opposed to pets? Because I mean, this is really a social disease, isn't it? I mean, can we call it a social disease? And how do you feel about this kind of reclassification?
Yeah, thanks for that question. I actually get it quite a bit. And there's two prongs to it, the way I perceive it. Um, so one of the things I do want to stress when we talk about this cat parasite that is zoonotic, meaning it can obviously transmit to people and other animals, and it thoroughly contaminates the environment, which sets up ecological battles between people who are in favor of like feral cats, and then people who are in favor of endangered species that Toxoplasma is wiping out because of these feral cats. So you raise an incredibly astute point that is hotly contested by multiple groups around the world. So in terms of human pets, most people who own a cat keep it indoors and they don't feed it raw or undercooked meat. And those cats tend not to ever have a toxoplasmosis infection and are considered relatively safe. And there's even studies that are done that show that cats provide an element of mental health, positive aspects of mental health that people capitalize on. So in no way do I want to batter cats or discourage people from having them as pets, but I do want to encourage them to do it safely. What I think is much more dangerous and much more important with respect to the environment is the feral cat problem. This is something that is, again, a major issue because no one likes to uh, collect feral cats and euthanize them in order to stop the spread of Toxoplasma into the environment. But this movement is gaining some strength because these oocysts persist for such a long time. And with climate change, they're increasingly found in oceans because of the runoff from the powerful storms we see. Toxoplasma has been found wiping out beluga whales, endangered species of dolphin, and this creates a very interesting dilemma. You know, do we suck it up and handle the feral cat problem, or do we say goodbye to these endangered species? So that is the issue.
But that's not one we're going to solve today, I'm guessing, but it does raise a big question.
Unfortunately not.
Yeah.
Unfortunately not. But
Well, I
you know,
have yet, I
yeah,
have
go
yet
ahead.
another reason to not like cats.
Yeah, I know there's a lot of cat lovers and cat haters, but I know there's a lot of cat lovers out there, and I want to assure them that there are safe ways to uh, continue enjoying your cat and all the joy that it brings.
Brandon, you turn on your camera. Go ahead.
Yeah, thanks for the talk, Bill. That was really great. I had a question. Could you talk a little bit more about your process? You said, when you're cultivating the toxoplasma and you're throwing all these drugs at it to see how it reacts, like the guanabans. How do you go about selecting those drugs? What's that process look like? Because it seems like, I would never think that a hypertension drug would react that way. So how did you stumble upon that?
Yeah, no, that's a great question, Brandon. I appreciate it. That is usually what I present as a seminar when I give research talks, because you're right. Why in the world would we pick an old blood pressure drug to work against toxoplasma? And that was not the logic behind it at all. So there is about 15 years worth of work that led up to that decision, and it involves looking at the mechanism by which the parasite switches from its growing form, its replicating form, into that latent cyst. So a multitude of students and postdocs in my lab through the years, in collaboration with Ron Weck here at IUSM, have found that a process called translational control, which is the process by which cells regulate how they make proteins and when, is very critical to the conversion of the growing form of the parasite to the latent form. Another group has found guanabans to be instrumental in one of the signaling processes that governs translational control. And that was the impetus for trying it in toxoplasma. We originally thought that the drug would keep the cyst locked into its latent form. So we were thinking it would be a prophylactic drug that HIV patients could take so that toxoplasma would not reactivate. It was a pleasant surprise to us to see that it not only blocked reactivation, but the reactivation was being blocked because the parasites were actually dying when they were hit with guanabans. And to this day, we don't know why that happens, but it is one of the things I'm trying to get a grant to elucidate.
Great, thank you.
So can you talk more about somebody was unmuting, it looked like, but can you talk more about the issue about screening that you said the United States has not, is nowhere in the United States is that been taken up, or is it, and how does that fit around the world? I'm just curious about it.
Sorry,
Yeah.
I was the unmuting person, and I was going to ask the same question. I'm in pediatric infectious diseases, and occasionally we get consulted to care for newborns with concern for a congenital infection, toxoplasmosis, being on the list. But we've been asked, like as a consulting team, why aren't we screening moms for this as well? And typically the answer is like, well, because there's nothing we would do for the mom. And it's unlikely that a baby will be born affected by this. So anyway, yeah, I would be very curious to know hear more about like what other countries are doing and why.
Yeah, so great. I can handle both of those questions. And Jimmy, I'm really glad you can provide that clinical insight because I think what you just outlined there is at the root of the misunderstanding that is currently plaguing the field. When I wrote that article for the conversation, I reached out to several pediatricians who are experts in congenital toxoplasmosis, Rima McLeod, Jose Montoya, and a few others. And they've been investigating this topic pretty much all their career, which is it's spanned several decades now. And most of the data that addresses the very concerns that you brought up is recent. I'm talking like since 2015. So it's only been in the last decade or so that a lot of these myths are beginning to be debunked regarding congenital toxoplasmosis. So the problems are we don't really have a good hand on the frequency at which this happens. We know toxoplasma is hugely prevalent. Okay, so that's not the issue. But old data suggests that the number of cases of congenital toxoplasmosis in the US is between 400 and 5,000 each year. So not a whole heck of a lot, but utterly devastating when it does happen. And many of these cases are not recorded because babies can be born asymptomatic. And then when they develop, when the congenital toxoplasmosis manifests months or years later, it doesn't get recorded. We also have absolutely no data on the number of spontaneous abortions or miscarriages or stillbirths that toxoplasma may be linked to. We do know that stillbirths are on the rise in this country, but we have no way without a surveillance program to gauge whether toxoplasma has something to do with that or not. So to give you some reference about other countries, France has been doing this toxoplasma surveillance program as a part of prenatal care for 40 years now, partly because the seroprevalence in France is 9 out of 10 people. So it's staggeringly common in France. And the speculation is they eat a lot of steak tartare, but I'm not quite sure what the real reason might be. In the States, the old estimates were about, 1 out of 4 or 1 out of 5 people. But again, without a toxoplasma surveillance program during pregnancy, we're really operating in the dark here. Now, why don't we do that surveillance when other countries like France, Estonia, Brazil started theirs in 2020? So if countries aren't doing it already, a lot of them do seem to come around. The US and the UK seem to be exceptions to this rule because I don't even think it's on the radar. And I'm trying to advocate for that along with Doctors Rima McLeod and Jose Montoya. The other issue that Jimmy raised is, well, there's nothing you can do. And that used to be true for a long time. That was the dogma. But newer study, I mean, it's hard to do clinical trials, and that was the other part, because you do not want to deprive a pregnant person of critical drugs that could save or minimize the damage. So it's very difficult to set up a clinical trial to address this. But in 2018, a study was released, a meta study that looked at this antibiotic called spiramycin. And this is administered when a woman seroconverts during pregnancy. And it's been shown to be incredibly efficacious at preventing transmission from mother to child if it's given early enough. If the parasite happens to breach the placenta, spiramycin doesn't do much good, and you can treat with antifolates. Studies that have shown that congenital cases treated with antifolates greatly reduce the severity of congenital toxoplasmosis. So the lack of treatment doesn't make any sense anymore because there's 2 arms of treatment that can be employed. Final issue that comes up is the cost-benefit ratio. These drugs that I'm describing, spiramycin, antifolates, they're very cheap. What about the cost of testing itself? That's been alleviated in recent years because Dr. Rima McLeod has developed a simple and very cheap finger prick assay that can be given to pregnant women very easily at like $4 a pop. There's also a saliva test that has been under development for a couple years now. Rima McLeod crunched the numbers. Okay, congenitally infected infants cost our country about $10 billion, okay, to take care of. The cost of a testing program would be less than $3 billion. So it's costing us more not to test than it would to implement this surveillance program.
That's super fascinating. Thanks a lot. I don't want to monopolize the conversation, but I do have a couple follow-ups to that, if it's okay.
Sure.
So first, you mentioned like spiramycin for moms who seroconvert during pregnancy. So I guess that implies that you would need a baseline test and then a follow-up test for either at some specified point or if a mom develops symptoms, as opposed to the way we typically screen for it, which is just with an IgG antibody, right? So I'm a little curious about like how that strategy would actually be implemented in a way that would allow us to provide treatment in like the clinical setting. And then I guess the related question is, are these like rapid finger stick tests? Like what are they actually testing for? If it's just for IgG antibody, again, like I don't know how that kind of helps us. And then the last thing I have is just a comment regarding the surveillance, which I agree, like sounds important. Could we accomplish that outside of like, doing clinical tests for a particular patient and instead maybe utilizing like dried blood spots that are collected, routinely for newborn screening?
Yeah, all excellent questions. And just so I don't belabor points that I already said, I'll address the first one first, about a basal test, a baseline test. So that would have to be done as soon as possible. So if you do that baseline test and the pregnant person is already showing antibodies to toxoplasmosis, odds are they've already been infected. And if that's the case, they don't need to be tested anymore because there's no reports that I'm aware of a person who's previously been infected passing it on to an unborn child. So in other words, they've already been vaccinated, and if they encounter the parasite in something they eat or in the litter box, it's not going to create enough parasitemia in order to transmit through the placenta. The mother's primed immune system will generate rapid antibodies and get rid of the infection. So a lot of women, approximately 1 out of 5, probably wouldn't even need continued testing because they already had Toxoplasma seropositivity before the pregnancy. But you're right, this is not like something you do once during pregnancy. This has to be monitored. And I've seen a couple different variations on this. Obviously, the more often you test, the earlier you can treat when you see a conversion. So that would be ideal. So I don't know what they do in France, but I think it's advocated that this would be part of a monthly or at least bi-monthly process to test pregnant people who showed no evidence of infection prior to the pregnancy. So I hope that hits on most of your points, Jimmy, but if you want to follow up in emails later, I'd be more than happy to do so.
Yeah, that was helpful. Thank you.
I'm sorry that there was one last thing I wanted to mention. We certainly can continue to educate people about the dangers of toxoplasma, and that would also greatly minimize transmission to pregnant people.
Bill, there's one quick question in the chat that just asks how long Toxoplasma gondii can live without a host before we take a pause. So how would you answer that? You see that question?
Oh, yes, I'm reading it now. Oh, well, this is another excellent question. And I think it depends on what stage of the life cycle you're talking about. So if it is like expelled out of the cat in the form of an oocyst, those oocysts are stable in the environment for at least a year. Some reports suggest that can be longer. So, those are sturdy containers and they can last a long time. And once it gets into a host, it presumably stays there for life. So I think the question is focused on how long can the oocyst persist in the environment? And again, that's like 1 or 2 years.
Well, I want to take a pause here and thank you for sharing with us today and raising lots of very interesting questions. We know that people have top of the hour things, so we like to take a pause here and say, if you have to leave, great. Thank you for joining us. Follow us along. You'll get a follow-up email with those links that we promised for the follow-up information. Join us for future events. But if you want to hang out and continue the conversation for a few minutes, we'll be here at least till the top of the hour because we can. But we did want to take this pause to allow those that really have to get going to do that. But join me in thanking Bill for sharing this information today and giving us a lot to think about.
Thank everyone for coming and for the excellent questions. I really appreciate it.
Well, great. And as we pause there, sometimes folks hang out, and this is a chance for us to just have some additional conversation. So does anybody that's still here have another question or a comment that you'd want to make sure we cover before we would move on.
Yeah, I have a question for Dr. Sullivan.
Go ahead.
So I've also heard that it's like a leading cause of like foodborne illness. So like, how does this connect to food safety?
Yes, you're absolutely right about that. Toxoplasma is one of the #1 food poisoning killers. So it's not one that it'll obviously make you sick, but it's also one that'll most commonly kill you if you get sick with it, usually because you get such a high dose of the parasites. There's two major ways you can pick it up from consuming foods. Unwashed fruits and vegetables can contain oocysts. So especially under cases of pregnancy, we tell people who garden to be especially careful of this. Cats like to use the garden as their bathroom, and potentially contaminated feces can get on fruits and vegetables that you grow. So wash the produce, cook meat, particularly lamb. Lamb seems to be one of the major culprits in the United States for transmission of toxoplasmosis. Many cases of congenital toxoplasmosis can be traced back to a recollection of having rare lamb. But other meats, even in the United States, can potentially contain toxoplasma tissue cysts, and those can be ingested. And another way is through raw shellfish. Like I said before, these oocysts are resistant to the salt water, and they can collect in shellfish like oysters or mussels. And when people eat those, those are filter feeding organisms, so they can collect tons and tons of those oocysts within the fleshy parts of the organism, and you're basically just inoculating yourself with a huge amount of oocysts, and it can be deadly.
That's not necessarily a pretty picture in my mind, but thanks for sharing.
No, it's not. And I'm sorry if anyone is a fan of raw oysters. Bad idea.
Yeah, I'm not a fan anyway. You solidified that. Are there other questions or comments?
I just remember one other thing to address Chelsea's point. In the United States, most of the toxoplasmosis food outbreaks that you see are related to game, wild game. Venison, bear was another one. I think that was in Canada. But people hunt and they don't sufficiently cook the animal that they ingest, and you'll see outbreaks of toxoplasmosis several times throughout the year related to game or hunting.
Well, I was particularly curious about, we may just have to talk more about the effects on behavior, which makes me, as a social worker, particularly one that focuses on early childhood, that made me curious about that. And your question of, do we have free will, or how does this impact behavior, that's very curious to me.
I share your interest in that topic. It's one of the things that fascinated me from the get-go.
Yeah. Well, and when we look around, it seems to me that there are I don't know what if the data would support it, but that there are increases in certain kinds of challenges and behaviors. And so what is that about is what I wonder. And now I wonder if this has some connection to that in some way, if it's toxoplasmosis or some other things, even in combination.
I used to tell my med students to look around at their neighbors because, if you count 4 or 5 people to the right or left of you, one of you has this parasite in your brain right now.
Right now.
And I tell them, based on the personality that they know, if they could guess who it might be or who owns a cat, or something like that.
If only we could just give them a test, we would know. Maybe that's part of your study you're trying to do. I don't know.
Well, you can certainly go get tested for toxoplasma.
Right.
I don't know if insurance will cover it, but tests do exist.
Yeah.
And the ones I was alluding to earlier specifically refer to cheaper testing that we could administer multiple times during a pregnancy.
Wow.
Any other questions?
Looks like Jerry is coming on. Okay, Jerry.
just curious. I like the question you didn't answer toward the end, and we didn't poll because you didn't have the poll feature about like if you could fix people whose personalities had been altered, would you? Which is an ethical question, right? And then I wondered, just in general, how people in your field deal with that. Like humans have been around a lot, and with cats and felines for a long time, and like what it means to be human without our parasites. Do people wrestle with that?
They certainly do. There's actually a whole body of literature out there that you can explore if you want, speculating that toxoplasma may have been instrumental in some societies or cultures in causing the culture to collectively take more risks and wage war and maybe dominate other cultures. So I don't know how much I buy into that. It's extremely impossible to test, but it's a fascinating line of thought that not only individuals, but maybe entire cultures have been influenced by this parasite in some way, shape, or form that has affected how human beings maybe spread around the world and how they treat one another.
Thanks.
Well, I think this is an excellent example of the kind of research that happens on our campus that is translational in our definition of generating and using knowledge to try to solve problems, and how it covers all kinds of different questions and aspects. So it isn't just a medicine thing. There's all kinds of implications and questions and directions that we might go, and why we all want to talk about it, why we all need to be together to have these discussions. So I can't thank you enough for being part of this and sharing with us today and leading us in this conversation to think about these things. So thank you so much, Bill.
It's my pleasure. And once again, I thank you so much for the invitation and the honor to be noted as a Tripp Scholar. It's a fascinating program, and I'm delighted to be a part of it.
Well, I hope you'll tell that to everybody and all your friends so that they join us too, because the more we have, the better off we are in sharing these opportunities. So thank you so much.
Thank you.